I'm a research scientist at LSE. I have been working on data assimilation with the Orchidea land surface model for about 15 years in order to improve its parameterization. And I have logically developed an interest in the potential of SIF for the task.
So I'm Fabienne Maignan. I'm also a senior researcher at LSE, and I've been working with the Orchidea land surface model like for 15 years now, and I'm interested in the processes like photosynthesis, phenology, and so in the recent years we have used proxy for GPP such as solar-induced fluorescence, but also the absorption of carbonyl sulfide. Thanks for the invitation. Today our talk is entitled Using SIF to Improve the GPP Simulated by a Land Surface Model. So you see here in this illustration mean annual fluxes integrated at global scale for the components of the global carbon cycle, that is atmosphere, land surfaces, and ocean. So in gray, you see the anthropogenic emissions, and you see that both land surfaces and ocean are currently acting as net sinks. The main uncertainty lies with the land surfaces. With this green arrow here, you see the net flux is estimated at 3.3 plus or minus zero point 0.8 gigaton of carbon per year. So this net flux results from two opposite gross fluxes of about the same magnitude, around 130 gigaton of carbon per year. The incoming flux is slightly larger. It represents the quantity of carbon that is assimilated through the photosynthesis process, and the opposite flux is related to respiratory processes. Today we will focus on this incoming flux, which is called gross primary production, or GPP. So why is net sink uncertain? So this net sink uncertainty results from the uncertainties of the gross fluxes. So GPP is very uncertain because first, it's a flux that exhibits a very large spatiotemporal variability. This is because the driving factors are also varying. They are the solar radiation, the temperature, the water availability. There are extreme events. You have different plants growing in different regions, on different soils. You have also often human management. And then GPP is not directly measured at scales larger than the leaf. So the resulting discrepancy in GPP estimates is illustrated with these maps where you see GPP annual means. On the top row, you have data-driven products, and below you have GPP simulated by land surface models. So you see different estimates in the tropics, but also in the mid to high latitudes. The GPP estimated at the global scale varies between 110 and 170 gigaton of carbon per year. You have another illustration below here where you see time series of GPP integrated over the tropics for various products and simulations. And again, you see a very large spread of about 50 gigaton of carbon per year. So we absolutely need to reduce this GPP uncertainty in land surface models to gain some confidence in climatic simulations. And to do this, we need to improve the process representation and use adequate observations to constrain these processes. So indeed, to understand and model the processes and calibrate their parameters, we rely on observations. On this first figure, you see that observations are available at various temporal and spatial scales. So starting from in situ flux estimate at flux towers to global observations with satellites. On this other illustration on the right, we have linked these observations to the processes. They may be more or less directly related to, like photosynthesis and transpiration, and so on. So today we focus on GPP and fluorescence observations. For GPP, we make use of GPP estimates at flux tower sites using the eddy covariance technique. You see here the map of the FluxNet networks. So we have a global distribution, but it's fairly uneven. At global scale, we benefit from GPP gridded products that are extrapolated this in situ information using machine learning methods. And we can cite the FluxCom product, for example. On the SIF side, and more generally fluorescence side, the in situ data sets are very sparse. We have data sets for active fluorescence measurements at UTLR, for example, and we have a few sites where we have SIF observations at canopy scale. But mainly we rely on satellite SIF products. So land surface modelers were very excited when they saw this first comparison between a map of the GOSAT-SIF products and a simulation by the BGC land surface model with a coefficient of determination of zero 0.8. Then we also add this scatter plot between GOSAT-SIF and the simulated GPP, where you saw a linear relationship between the two variables with different slopes between different biomes illustrated by different color. Then you have these mean seasonal cycles over decidual broadleaf and evergreen needleleaf forests in the boreal region, where you see a very close relationship between GPP in blue and GOMI2-SIF in magenta, and you have a different seasonal cycle for two NDVI products in green and in orange. We can try to explain the close relationship between GPP and SIF at canopy scale using this simple light use efficiency model, where you see both GPP and SIF are proportional to EPAR, which is a quantity of absorbed photosynthetically active radiation. And you see that for SIF, we have this epsilon F, the escape factor, which explain why the relationship between GPP and SIF depends on the canopy structure, and that therefore on the biome. So here in this table, you see the main SIF products with characteristics. And below we have an illustration over the Nile. So in the center, we have the GOMI2 product with a very large footprint of 40 kilometers. On the right, you see the OCO2 image. So the footprint is much lower, but this is a thin swath, and then it delivers a poor spatial coverage. On the contrary, on the left, you have the image for Tropomi with a 3 by 5 square kilometers and a large swath answering daily global coverage. So now let's look at the tools that we are using for our research, and we start with the ORCID land surface model. So this model can be driven by meteorological fields, or it can be coupled to an atmospheric model. Below you have a very schematic representation of the model with fast processes that are resolved at a half-hourly time step, and below other carbon processes that are treated at a daily time step. The vegetation is prescribed using maps of plant functional types. So a plant functional type groups together plant species that have a similar morphology and physiology under the same climatic conditions. So let's have a brief look at the main process driving GPP, that is photosynthesis. So in some leaf cells, you find chloroplasts. Inside these chloroplasts, you have thylakoid membranes where there are photosystem 2 and photosystem 1 that have chlorophyll pigments which can absorb the sunlight energy. And this drives an electron transport chain, which in fine produces energy for the dark phase of photosynthesis, where the carbon fixation happens and sugars are produced. In the land surface models, you mainly found for photosynthesis the Farquhar and Collatz models. The photosynthesis model is coupled to a stomatal conductance model, and we also have a fixed law of diffusion through this series of conductances here, from the conductances for the leaf boundary layer to the stomatal conductance and the mesophyll conductance. And these systems of linked equation is solved for each canopy layer, and then you integrate all assimilation across this canopy layer and you compute the GPP. And then I will leave the floor to Cédric, who will continue with the data assimilation framework, and he will present our main results.
Yes, I will start by providing an overview of how we are using SIF data in order to optimize the simulation by the Orchidea model. So we rely on a data assimilation system whose principles are illustrated on this schematic. So it is based on the Bayesian framework in order to minimize iteratively a misfit function that quantifies the mismatch between the model simulation and some data, and also the departure from some prior information of the optimized parameters. So the two terms of this equation are also weighted by the covariance matrices associated to observations and parameter. But in order to assimilate SIF data, we first have to develop a so-called observation operator in order to simulate the variation of SIF at the top of canopy with the Orchidea model that can be comparable to some spaceborne or in situ observation. So the development of this SIF observation operator is still ongoing. We started with a very simple linear relationship between SIF and GPP, and now we are moving towards more process-based oriented modeling from the leaf scale to the canopy scale, and I will discuss that a bit more in the next slide. In our data assimilation framework, we optimize parameters related to photosynthesis, phenology, and also to the SIF model. The calibration is performed PFT per PFT, and mostly we assimilate SIF data from different spaceborne instruments, GOM2 first, TOCO2, and now TROPOMI data, at a quite coarse spatial and temporal resolution. And to perform those assimilation, we select first handful pixel, which are selected for their homogeneity with respect to the targeted PFT. And now we're also moving towards co-assimilation of SIF and GPP. And for the GPP product, we consider mostly the in situ data from FluxNet sites, for instance. So this is an illustration of our first work, considering a simple linear relationship between SIF and GPP. So we have optimized about 15 PFT-dependent parameters, again related to phenology, photosynthesis, as well as the slope and offset of this relationship. So we assimilated 4 years of GOM2 data, and this figure illustrates the positive impact that the assimilation of SIF has on the simulation of GPP at the global scale. So those two maps compare the prior simulation on the left, the posterior simulation in the middle, so after assimilation of SIF data, and the difference between the two. Mostly, if you look at the right map, you see that most of the regions are in blue, which indicates a decrease of GPP after assimilation. At the global scale, the reduction of GPP is about 30 gigaton of carbon per year. So since then, we have also developed more process-based modeling of SIF in order to better represent and also exploit the distinct temporal variation of the concurrent photosynthesis and fluorescence processes. So for the top of the canopy, we first apply the formalism that was developed by the scope model at the leaf scale and applied it at the top of the canopy. And in this formalism, we calculate SIF emission from the top of canopy for the two photosystems, and the SIF emission by photosystem 2 is corrected by a regulatory factor which depend on environmental conditions. So we've developed some parametric model in order to simulate SIF for the two photosystems based on simulation by the scope model. And the regulatory factor depends on the quantum leap of fluorescence, which is calculated by Orchidea as a function of the quantum leap of photochemistry, which has been derived by the photochemistry module in Orchidea that was already implemented, and also the relative weight constant of photoprotection. And because no physical model were existing at that time, we have developed our own parametric model fitted on leaf scale fluorescence data. Using this model, we have been able to assimilate SIF data from the OCO2 instrument, again at coarse spatial and temporal resolution. The figure on the left shows you the impact, again, of the positive impact that the assimilation of SIF has on GPP. The figure shows the temporal variation of the global scale GPP budget, and you can see a large decrease, yearly budget between the prior simulation in blue to the posterior simulation in orange by, again, 30 gigaton per year. And you can see that the optimized simulation are in better agreement with data-driven product, Fluxcom and Fluxat, but in particular with respect to Fluxat data. However, this overall agreement hides some PFT discrepancies. This is illustrated on the figure on the right, which shows the mean seasonal cycle of GPP estimated by Fluxcom in black compared to the model simulation, the prior in blue and the posterior in orange. So overall, we observe a general improvement of the simulated seasonal cycle in terms of magnitude and seasonal variation for most of the PFT, but still we observe a degradation for 4 PFT out of 14. As for instance, the one that is shown here in red, temperate deciduous broadleaf forest, you can see here that the posterior simulation overestimate largely the mean seasonal cycle. So there are still some challenges that we need to resolve. So in order to still improve the way we are able to simulate the spatial and temporal variation of SIF and GPP with the Orchidea model, we are now working on a two-flux radiative transfer model. And now we are able to not only simulate the regulatory factor, as in the formalities that I have presented earlier, but now we simulate the SIF emission at the leaf scale, following the concept of the fluor mod leaf model, which is scaled up to the top of the canopy using a two-stream scheme. And we now also account for direct and diffuse radiation regime within the canopy. So we expect that with this new scheme, we will be able to improve our simulation of both SIF and GPP. And we are, with this new scheme, investigating the benefit of co-assimilating SIF and GPP data. And now we consider the spaceborne SIF data from the TROPOMI instrument, and in particular the TROPOSIF product, because as Fabienne showed you earlier, of the higher spatial and temporal coverage provided by TROPOMI data. I will not go too much into the details, but the figure on the right show you our preliminary results with a not consolidated version of this two-flux radiative transfer model, in which we have co-assimilated GPP and SIF data. And you can see a large decrease of the prior simulation here in blue. And the optimized simulation are now in better agreement with other land surface model simulation and data-driven estimates. But I will go a bit in more detail on that in the next slide. Here we focus now on the constraint brought by SIF data only on simulated GPP. So we have assimilated 3 years of Tropo SIF data over about 20 selected pixels at higher spatial resolution and higher temporal resolution than with OCO2 and GOM2. And you can see on those box plot the RMSD with respect to SIF and the RMSD with respect to GPP over those pixels and comparing the prior simulation in a range to the optimized simulation. So it was expected, you can see with respect to SIF, that after assimilation of SIF, the RMSD after optimization is lower than before optimization. But for most of the PFT, you have the same improvement with respect to GPP, which is shown on the figure on the right, except for 3 PFT for now, for which you can observe a degradation of the simulated GPP with respect to the FluxSat and FluxCom data-driven estimates. So this comes from apart from uncertainty in the a priori parameter of the new SIF model and some parameter interactions during the optimization process. So this is why we believe that co-assimilating SIF and GPP will improve the simulation of both variables. So this is illustrated on the next slide for the ideal case of boreal needle-leaf evergreen forest. For this analysis, we have conducted 3 different data assimilation experiments. The first one, we assimilated only TropoMe SIF data. In the second experiment, we have assimilated only in situ GPP data. And the third experiment, we assimilated jointly SIF and in situ GPP data. And these figures show you the mean seasonal cycle over independent sites and independent pixels with respect to SIF on the left and GPP on the right. So what you see is that logically, after assimilation of SIF, you have a strong impact, positive impact of SIF, but also on GPP. And what is shown here is that with respect to data-driven product, the simulation of GPP after the assimilation of SIF data only provides a better model data agreement between Orchidea simulation and data-driven estimates. At the side scale, the results when you assimilate only SIF data is just slightly lower than when you assimilate only a GPP data. But again, this is an ideal case, and it's not always the same for all other PFT, and there are other challenges that we still need to resolve.
So this is what we plan to do in the coming years, thanks to several funded projects. So first, we have a French ANR project where we want to study the photosynthesis downregulation in abiotic stress condition. So the first step is for us to derive a new NPQ model that is also valid, for example, for drought conditions. To do this, we use measurements of active fluorescence, and we have started with Arabidopsis thaliana of wild types and NPQ mutants, and also with poplar. And now at the lab, we have David Martini, who came with the OstroSIF database, which is a database with active and passive fluorescence measurement across 7 sites in Europe. And we have also started doing some statistical analysis using XGBoost or random forest model and Shipley values to identify the driving factors of NPQ in various environmental conditions. Then we also plan to replace within Orchidea our simple linear water stress function by a complete hydraulic architecture. And this architecture gives us access to simulated leaf water potential. And then it will allow us to explore the interest of using remote sensing vegetation optical depth. So it's a variable that is acquired in the microwave domain. And for the lowest wavelengths, the VOD is proportional to the canopy water content, and we can have a simple model based on the LAI and leaf water potential. So we could co-assimilate SIF and VOD. Then we have another French project where we plan to implement our fluorescence model in a version of Orchidea that integrates an explicit representation of the nitrogen cycle. Then, with this new Orchidea version, we can better study the nitrogen use efficiency under various conditions based on fluorescence measurements, and also we may look at satellite products of leaf chlorophyll content. The idea is to better know where is the nitrogen within the leaf, which proportion can we find in Rubisco, which is the enzyme that catalyzes carboxylation, and which quantity we find in the chlorophyll pigments that are used for the capture of sunlight energy. And finally, so David Martini had a successful postdoctoral fellowship from the Marie Sklodowska-Curie Action. And we will replace in the Orchidea model the Farquhar model with like an extended version following the model of Johnson and Berry, where the electron transport is also represented in a more mechanistic way. And then you have the photosynthetic control that will regulate the functioning of cytochrome B6F and activate non-photochemical quenching to balance energy excitation between the two photosystems. And this model also produces NPQ and SIF. So it's time to conclude now. So what you can retain regarding our modeling and data assimilation frameworks is we favor co-assimilation of SIF and GPP to limit model overfitting. We still have work to do because we still have some results for some PFTs that we don't understand. They may be related to SIF prior values and the authorized range of variation of the parameters during the data assimilation phase. It would be nice to have a large network of in situ SIF observations, like an equivalent of FluxNet for GPP. So we now believe that benefits of sparse-borne SIF data compared to in situ GPP data is mostly for undersample PFT where you have no in situ flux tower, for example. So regarding the upcoming Flex mission, so on the bright side, we will have a high spatial resolution of 300 meters that will be more consistent with the one of the eddy covariance flux data. For us, it will be easier to have pure plant functional type pixels. And we are also happy to have complementary information from Sentinel-3, for example, regarding the chlorophyll content that could be co-assimilated with SIF or even NPQ information coming from Flex. So there are still some limitations. So the low visit frequency is a problem for us, and we are not yet sure about the retrieval error. Plus, given that there is no high spatial resolution meteorological forcing available, we still probably need to perform some data binning of Flex for our research study.